Perforin, also called cytolysine or lymphocyte-performing protein, is a protein located in the gene 10 in the chromosome Q.22.1. Q means the Q, that is the long arm of the chromosome. Its function is to form calcium-dependent pores of 20 nanometers in other cells' membrane, if they are infected. This protein is secreted as a monomer, that means individually, but they must join to perform their function. It is made up of 555 amino acids and stored in the cytoplasmic granules of the cytolytic T lymphocytes. T lymphocytes are an essential part of the immune system, and they are formed in the bone marrow and matured in the thymus. They are divided in 4 different types: cytotoxic T cells, helper T cells, regulatory T cells, and natural killer cells. Cytotoxic T cells have a co-receptor called CD8. This co-receptor partners with the T cell receptor and MHC class I molecules to form a sort of bridge. This bridge allows the cell to recognize if the other cell is infected. If so, it will secrete perforin to kill the cell, avoiding the spread of the pathogen. Helper T cells have a different co-receptor called CD4. The CD4 partners with the receptor of the cell and with MHC class II molecules to analyze cells. If they find a peptide of an antigen, they would produce a molecule called cytokines to produce signals to the other immune cells, and it would signal mainly helper T cells and B lymphocytes to produce antibodies. Peptides are chains of amino acids found in nature that perform a lot of functions of life. Then, regulatory T cells also have CD4 in their surface, but they are used in other way. Their function is to stop the immune response when it's no longer needed, obviously. This cell can stop the immune response in several ways: by killing activated immune cells, releasing cytokines that suppress the immune response, or changing the way dendritic cells behave so that they can't activate T cells. Dendritic cells analyze antigens and present it to T cells for them to act. Natural killer cells, or NK cells, are a component of innate immunity. This makes them very different from other T lymphocytes because they are all part of the adaptive immunity. Innate immunity is the first line of defense against the pathogen, and it's a series of chemical, physical, and cellular defenses against the pathogen for avoiding its spread. Adaptive immunity is the second line of defense, and this defense attacks directly to the pathogen but can make errors and produce an autoimmune disease. Returning to NK cells, they produce apoptosis. Apoptosis is a controlled way of cellular killing that is essential for the correct development of bodies. Some diseases related to perforin are immunodeficiency syndrome, hemophagocytic lymphohistiocytosis syndrome, and the deficiency of PRF1. Immunodeficiency syndrome is a disease caused by the defects, rather inherited or acquired, that produce the malfunctioning of T lymphocytes and NK cells that won't produce enough perforin. These may result in constant infections. Then, hemophagocytic lymphohistiocytosis syndrome is a disease caused by the substitution of 2 amino acids. This produces an excessive amount of immune cells, and that may produce symptoms like fever, spleen, or enlarged liver. Then, the deficiency of PRF1 is a disease that presents a disorder in the immune regulation caused by the complete loss of T cytotoxic lymphocytes. And that's the end.